Hello class, welcome back to your botany class. So today I would like to discuss with you the endomembrane system of the cell. In the previous class we have discussed about the plasma membrane, how a plasma membrane looks like in the outer structure, then the structure of mitochondria, the functions of it, then chloroplast, the functions of it. These are the cell organelles that we have discussed, and so today I would like to discuss with you the endomembrane system of the cell. So these cells, organelles, particularly endoplasmic reticulum and Golgi complex and lysosomes, they form the endomembrane system of the cell. Okay, so firstly, let's see the endoplasmic reticulum. The term endoplasmic reticulum was coined by Porter, and they are absent in prokaryotic cells, and all prokaryotic cells, they are devoid of all cell organelles, and they are also absent in mature mammalian RBC, red blood cells. Okay, so this endoplasmic reticulum, there are double membrane cell organelles, and the membrane of this cell organelle, they resemble the plasma membrane. It consists of proteins and lipids, and the structure, it resembles the plasma membrane. And this endoplasmic reticulum, they are found attached to the nuclear membrane. They are found attached to the nuclear membrane, and of course, there are some another type of endoplasmic reticulum, they remains in diffuse state or in the peripheral region. Okay, so this endoplasmic reticulum, they exist in three morphological forms. They exist in three morphological forms: the cisternae, which are elongated sac-like structures, and they have a thickness of about 4 to 5 nanometer. And the another form is the vesicles. They are 2.5 to 50 nanometer in diameter, and they are oval or roundish or spherical in shape. And the other morphological form is the tubules. And these tubules are tube-like structures. They are branched, elongated tube-like structures, and they have a diameter of about 5 to 10 nanometers. So these cisternae, vesicles, and tubules are the three morphological forms of endoplasmic reticulum. So these endoplasmic reticulum are of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum, on the basis of the presence or absence of ribosomes on its surface. So when ribosomes are present on the surface of endoplasmic reticulum, that type of endoplasmic reticulum is known as rough endoplasmic reticulum, and those endoplasmic reticulum which are devoid of ribosomes, they are known as smooth endoplasmic reticulum. And usually the rough endoplasmic reticulum, they are found attached to the nuclear envelope, while the smooth endoplasmic reticulum, they are found towards the periphery of the cytoplasm. Okay, so this rough endoplasmic reticulum, due to the presence of ribosomes, they are the sites of protein synthesis, or they are associated with protein synthesis. And smooth endoplasmic reticulum, they are involved in lipids and steroid synthesis. As such, they are found primarily in the glandular cells. Okay, so these are the two types, rough and smooth. And these ribosomes, we have discussed, it consists of two subunits: large subunit, small subunit. And this, in eukaryotes, the ribosomes are attached to the endoplasmic reticulum by the large subunit, the 60S, and with the help of glycoproteins called ribophorins. So these ribosomes attached to the endoplasmic reticulum by the 60S with the help of glycoproteins. We have discussed about glycoproteins, and this type of the type of glycoproteins are called ribophorins. Okay, so what are the functions of endoplasmic reticulum? The main function is to provide mechanical support. Since it forms the main endomembrane system, it provides mechanical support to the cell, and it forms a sort of intracellular skeleton. It forms the main framework. Then the next function is intracellular transport. Okay, polypeptides and raw proteins are which are synthesized from the ribosomes. They are transferred to the endoplasmic reticulum from the ER. They are transferred, polypeptide and raw proteins which are synthesized on the ribosomes to on the ER, they are transferred to Golgi complex to be transferred to other parts of the cell. In the Golgi complex, they undergo modification, and as such, they are transferred to other parts of the cell. And next is this ER, they are associated with the synthesis of lipids and steroids. And this smooth endoplasmic reticulum, the membrane have an enzyme, and these enzymes helps in the detoxification of substances, foreign substances or toxic chemicals or substances that enters into the cell. When they enter into the cell, these enzymes which are present on the membrane of smooth endoplasmic reticulum are activated. As such, detoxification of those toxic substances take place, and as such it's eliminated. Okay, then next is the formation of microbodies like peroxisomes and lysosomes are formed by the endoplasmic reticulum. Then the another cell organelle is the Golgi complex. This Golgi complex, in case of animals, they are in case of animal cells, they are localized near the nucleus, near the endoplasmic reticulum, while in case of plant cells, they remain in dispersed or diffuse states, and they are known as dictyosomes. Okay, the term Golgi complex was first given by Camillo Golgi in 1898, and he observed or he discovered it in the nerve cells of barn owl and cat. Okay. So like endoplasmic reticulum, this Golgi complex, they also consist of, exist in three morphological forms: the cisternae, vesicles, and tubules. These cisternae are sac-like structures, flattened sac-like structures, and the vesicles, they are formed by the breaking up of the cisternae. They are oval, spherical, and they contain secretory enzymes. And tubules, they are tubeless structures, and they are associated with the cisternae, forming fenestrated plate. And the Golgi complex or the dictyosomes, they have polarity. They exhibit polarity. They have a polarity, and the two phases are the convex or cis or forming phase, or it's also known as the proximal phase. And the other phase of the Golgi complex is known as the concave phase or trans phase or maturing phase or the distal phase. So this convex phase is known as the receiving end of Golgi complex, and the concave phase is known as the discharging end of Golgi complex. So what are the functions of Golgi complex? The first function is the glycosidation of lipids and proteins. That means addition of carbohydrate to lipids and proteins. So this addition of carbohydrate to lipids is known as glycosidation, and it results in the formation of glycolipids, and addition of carbohydrate to protein forms glycoproteins, and this takes place in the Golgi complex. And we know, we've seen the function of these glycoproteins and glycolipids in plasma membrane. They serve as receptors, cell recognition, acceptance or rejection of cells and tissues during organ transplant. So glycoproteins and glycolipids, they are formed in Golgi complex. Next is they store, condense, and packaging of materials. Then they also form secretory vessels and secretory vesicles. Then another function is formation of lysosomes, and they are involved in formation of acrosome. Acrosome is the tip of the sperm, and which secrete an enzyme known as lysine, which dissolves the wall of the ovum during fertilization. So this Golgi complex is the main assembling factory of the cell, where the different raw materials, they are assembled to get a final conformation. The main assembling factory of the cell, where the different raw materials, they are assembled to get a final conformation. And the next cell organelle is the lysosome. So these lysosomes are single-walled, and they were first discovered by Christian D. Duvet in 1955. They are present in eukaryotic cells while they are absent in bacteria. Okay, so these lysosomes, they are also known as suicidal bags. They are also known as suicidal bags, and they exhibit polymorphism. The lysosomes exhibit polymorphism. Why? What is polymorphism? Is that they can exist in different forms. They can exist in different forms, and okay, so the different forms are primary lysosome or lysosome, then heterophagosome, residual body, and autophagosome. Okay, so this primary lysosome, these are the vesicles, or these are the first form lysosomes containing hydrolytic enzymes, and they are formed from the Golgi complex. These are formed from the Golgi complex, and they form the first type of, or the primary type of lysosome. Okay, so now the second type of lysosome, or the secondary lysosomes, are heterophagosome, residual body, and autophagosome. What is heterophagosome? Whenever a cell comes in contact with any foreign or external material, it can be solid, or ingestion of the solid is known as phagocytosis, or ingestion of the liquid is known as pinocytosis. So this ingestion or engulfing of the solid material forms the food vehicle. It forms the food vehicle by the cell, and whenever this food vehicle comes in contact with lysosomes, that structure is known as heterophagosome, and it is concerned with the digestion of the food vehicle. This heterophagosome is concerned with the, or which consists of the food vehicle and lysosome containing hydrolytic enzymes, is concerned with the digestion of this external or food material. So heterophagosome. Then the next type of lysosome is the residual body. Okay, this heterophagosome is also known as digestive vehicle. Next is the residual body. So the undigested material. Since lysosomes, they contain hydrolytic enzymes, they break down this food vehicle and absorb it at the residual body, or the remaining undigested material, they forms the residual body, which are eliminated in the end. They are eliminated outside the cell. Okay, then the next type of lysosome is the autophagosome. Autophagosome, or cell eating. Okay, whenever the cell is hungry or is in need of energy, these lysosomes can digest a part of its own cell. It can digest some of the cell organelles or a part of the cell. Any content that is a part of the cell. So it can be mitochondria, it can be chloroplast, or it can be even Golgi complex, or even any other part of the cell. So lysosome digesting a part of the cell or eating up a part of its own cell is known as autophagosome, or it's also known as autophagic vehicle. And since it can digest a part of its own cell, it is also known as suicidal bags, killing itself. Okay, so similarly, these undigested remains, they form the residual body, and they are eventually eliminated out of the cell. So these are the four types of lysosome. Primary lysosome, heterophagosome, residual body, and autophagosome are the four types of lysosomes. Okay, what are the functions of lysosomes? Okay, one is the intracellular digestion. As we've discussed, these lysosomes, they are concerned with the digestion of a part of its own cell. As such, it takes part in the cleaning up of the cell or providing energy to the cell. So intracellular digestion, then extracellular digestion. We've discussed that engulfing of the external material or food material or liquid by phagocytosis or pinocytosis is finally digested by the lysosomes by releasing the hydrolytic enzymes. So in extracellular digestion. And another function of lysosome is metamorphosis. Okay, metamorphosis, development. It is a term of development. So these lysosomes play an important role in the development of animals by digesting the larval parts, the young parts, and helping them to develop into adult ones. So by releasing enzymes during the process of metamorphosis. Then another function is during fertilization. Okay, we've discussed that this Golgi complex they play an important role in the formation of acrosome of sperm. Okay, so this sperm, the acrosome of the sperm contains a particular enzyme known as lysine, and this lysine enzyme helps in the digestion of the cell wall of the ovum, of the wall of the ovum, and that is secreted, is due to digestion of the wall of the ovum is due to the presence of lysosomic enzymes. Okay. Then another function is seed germination. In plants, lysosomes helps in the germination of the seed. And we know that there's the blue bee they are able to fight the foreign substances or any other external substances that or antigens that enter into body. That is because of the lysosomic activity of the WBC. They can act against these bacteria or virus or any other pathogens that enter into our body. That is because of the lysosomic activity of WB cell. So intracellular digestion, extracellular digestion, metamorphosis, fertilization, seed germination. These are, and even the actions of the WBC against the foreign substances, these are some of the important functions of lysosomes. So these three cell organelles, endoplasmic reticulum, Golgi complex and lysosomes, they form together, they form the endomembrane system, and they work in a coordinated manner. They do not work in isolation, but they work in a coordinated manner, and they form a sort of intracellular skeleton, and thereby providing mechanical support, particularly the endoplasmic reticulum. And this endoplasmic reticulum and Golgi complex, they exist in three morphological forms. In both the cases, cisternae, vesicles, and tubules. And cisternae are flattened sac-like structures. Vesicles are oval spherical, and tubules are tube-like structures, and they exist both in endoplasmic reticulum and even in Golgi complex. And rough endoplasmic reticulum, which are associated with the ribosomes, they take active part in protein synthesis, and as such, they are found in abundance in those cells that are involved in protein synthesis. While smooth endoplasmic reticulum is associated with lipid and steroid synthesis, and they are found particularly chiefly in the glandular cells. And of course, the smooth endoplasmic reticulum, they have detoxifying enzymes on its membrane. As such, they help in detoxification of substances that enters the cells. And these ribosomes are attached to the ER by means of the larger subunit, and the substances or the polypeptides and proteins that are synthesized in the endoplasmic reticulum, particularly rough endoplasmic reticulum, they are transferred into the Golgi complex, where they undergo modification, to get a final conformation. As such, Golgi complex is also known as the assembling factory of the cell, where different materials are assembled to get a final conformation. And glycosidation, that means formation of glycoproteins and glycolipids, which act as receptors on the cell membranes. They are formed in the Golgi complex. And then this Golgi complex, they are responsible for storing, condensing, and packaging of materials and transporting them to other parts of the cell. It's also another important function of Golgi complex. Then besides this, they form the secretory vesicles, or they form even the lysosomes, which contains hydrolytic enzymes, which are released during the process of digestion, and they take part in the formation of a chromosome. So lysosomes, intracellular digestion, extracellular digestion, metamorphosis, then fertilization, seed germination. These are some of the important functions of lysosomes. So these three cell organelles, they work in coordinated manner. Of course, not all the cell organelles, they do not function in isolation. All the cell organelles, they function in a coordinated manner, and the function of one is related to the other. So in unicellular organisms, where the single cell function perform all the cellular activities, while in case of multicellular organisms, there's division of labor. However, the cells, even in case of the cell organelles, even in case of multicellular organisms, they work in coordination, and these cell organelles are absent in case of prokaryotic cells, and they are found only in eukaryotic cells. So with this, I wind up today's class, and in the next class, we'll see more of the nucleus on which the endoplasmic reticulum are attached, and I hope to see you again. Thank you.